Hello, and welcome to the Rejuvenation Roundup podcast, where we explore the latest in life extension and anti-aging science with a dive into a month's worth of insights and new breakthroughs. This podcast is a combined effort of the Lifespan Extension Advocacy Foundation, which operates Lifespan.io, and Future Grind, a podcast that explores the ethics and impact of emerging science and technology. March marks the beginning of spring and the time for renewal, and researchers around the world have been hard at work. Here's what they've been up to. Starting off with our research roundup. An accepted manuscript in eLife Sciences has described eye age, a new clock that uses deep learning to analyze the eye in detail in order to predict chronological age and age acceleration. This project was developed with the knowledge that fundus imagery, which shows the blood vessels at the back of the eye, can be a useful diagnostic tool for eye diseases like age-related macular degeneration and diabetic retinopathy, along with seemingly unrelated diseases, such as hypertension and cancerous tumors in entirely different body parts. The researchers hold that this diagnostic power is because such diseases cause tiny changes in the vascular system that show up in the smallest capillaries first, and retinal capillaries are particularly small. However, detecting these tiny changes is difficult for human beings, even with modern instruments. Therefore, the researchers have employed artificial intelligence, using a deep learning system to build their aging clock. This study drew its data from a database using images from over 100,000 people for development and about a quarter that many for tuning. To test the robustness of their model, the researchers turned to the UK Biobank, which contained nearly 120,000 relevant images and different demographics. The researchers used eye age data to create an age acceleration measurement, eye age accel, similar to acceleration measurements in other clocks, and compared this measurement to UK Biobank genomics through a genome-wide association study, looking for the genes that were strongly associated with rapid eye aging. As expected, many of the associated genes were directly linked to the eyes and associated diseases. Some of the other genes were linked to cancer development and hearing loss. One particular gene, which has been shown to be associated with shorter lifespans, was of special concern. Inhibiting it in these flies was found to increase their lifespans. The researchers also noted that their work confirms previous research showing a human allele that decreases the expression of this and other negatively associated genes in this study, marking a prospective target for potential future therapies. As a visual biomarker that only needs imagery as an input, eye age may be a simple tool for researchers and clinicians to measure accelerated aging and gain valuable information about the prognosis of eye diseases. Future research using this clock alongside other clocks will determine its value in measuring the effectiveness of interventions against age-related diseases. Scientists have shown that a protein called growth differentiation factor 11, or GDF11, can reverse depression-like symptoms in naturally aged mice and in a mouse model of depression. Severe mental disorders, including depression, have been linked to significantly shorter lifespans. According to one study, depressed people have a higher incidence of various diseases of aging, such as cardiovascular and cerebrovascular diseases, metabolic syndrome, and dementia. This makes depression an important target of longevity research, but treating depression is difficult. In this new study, published in Nature Aging, the researchers investigated GDF11 as a potential antidepressant. This protein plays an important role in the development of the central nervous system and regulates neurogenesis in adults. Depression has been linked to a decrease in the volume of the hippocampus, the main locus where adult neurogenesis occurs. Previous research has shown that supplementation of this protein can increase hippocampal neurogenesis in aged mice. In the study, naturally aged mice showed a significant increase in depression-like symptoms compared to young controls, but treatment restored many of the measured parameters to youthful levels. As expected, older mice also experienced a decline in cognitive abilities. But this cognitive decline was reversed to youthful levels by the treatment. The researchers then investigated the mica's brains. Analysis of neurogenesis markers revealed a 53% increase in immature neuroblasts in the subgranular zone, the part of the hippocampus heavily populated by neuronal stem cells, indicating improved neurogenesis. The treatment also decreased levels of cellular senescence in the subgranular zone almost to the levels observed in young controls. In cultured hippocampal neurons, theoprotein caused a significant increase in autophagy and neuronal activity. The researchers also experimented with young mice in which depression-like behavior was induced. Just like with naturally aged mice, the protein alleviated most depression-like symptoms. The link between depression and aging calls for a serious investigation. This study suggests that the relationship between them might be mediated by decreased hippocampal neurogenesis, which can be attenuated by systemic administration of GDF11. Lower levels of this protein in depressed young adults lend some support to this hypothesis. In a new study published in Aging Cell, researchers have shown that two promising anti-aging agents, the antibiotic Rapamycin and the anti-diabetic drug Metformin, reverse aging in a population of intestinal stem cells. The function and structure of the intestinal epithelium, a single cell layer that lines the small intestine and colon, is maintained by the residing stem cells. Intestinal stem cells continuously divide to generate several types of progenitor cells. Stem cell exhaustion is a hallmark of aging, and reversing it requires an understanding of the transcriptional and metabolic changes that intestinal stem cells undergo with age. In this study, the researchers managed to bring aging intestinal stem cells to a more youthful state by treating them with rapamycin and metformin. The researchers used several groups of mice for the experiments. First, they harvested intestinal stem cells from young and old mice. Prior to tissue collection, the mice were fed a purified controlled diet for 3 months. The researchers then performed single-cell RNA sequencing and showed that the transcriptomic profiles of both stem cells and their progenitors changed dramatically with age. Further analysis showed that several metabolic pathways were perturbed in the stem cells of old mice. Things like fatty acid metabolism, glycolysis, and ribosome pathway were upregulated, while cell cycle pathways were suppressed. These changes reduced the proliferative capacity of the cells. By applying bioinformatics tools, the researchers revealed that age-related transcriptional alterations of stem cells had been passed on to the progenitor cells. Next, the researchers analyzed intestinal stem cells collected from 2 groups of old mice receiving diets, supplemented with either metformin or rapamycin. The transcriptional profiles of the cells taken from the old mice treated with either one of the drugs were significantly more similar to the young animals. Moreover, both drugs restored the proliferative capacity of the cells and reversed some of the metabolic changes observed in the old cells. Interestingly, metformin appeared superior to rapamycin for stem cell rejuvenation in a number of aspects. First, it reversed age-related deficits in fatty acid metabolism and glycolysis, while rapamycin did not. Second, metformin showed a greater effect than rapamycin in reversing perturbed oxidative phosphorylation in the main stem cells, as labeled by the study's software. Finally, rapamycin was less efficient than metformin at restoring the developmental trajectory of the intestinal cells by speeding up cell maturation delayed by aging. This study revealed the stem cell reprogramming abilities of therapeutic agents that are well known for their anti-aging abilities in animal models. Aging stem cells are a major obstacle to organismal rejuvenation. Drug repurposing, if effective in humans, is an attractive approach to targeting it. That's it for our research roundup. You can find more on these and other stories on our website at lifespan.io/roundup. In March, Life Noggin released 4 new videos, including one focusing on how your body changes as you age. Here's some of that.
Welcome to Build a Body Workshop. When making a human, you have to remember to stuff it with blood, or it doesn't work. Give it the ability to remember every lyric from Paramore's After Laughter album, but not how to do basic math. Give it ADHD, and we'll throw in anxiety and depression at no extra charge. Now just add the heart, which is admittedly a little defective, and voila, you have a human named Pat.
How to make friends at 30 years old. I can't?
But what happens as Pat ages? Let's see how things change as you humans get older. Hey there! Welcome to Life Noggin. After a couple of decades of growth and development, you humans reach your maximum amount of bone and muscle mass. Your 20s are also the time when your brain finishes developing. Menstruation becomes more regular. You may also acquire adult acne. And you have the highest chance of developing immune system disorders like lupus or rheumatoid arthritis. In your 30s and 40s, things start to decline. Yay! You lose bone mass, which can make you shorter and more susceptible to injuries. You start losing muscles and getting weaker, a condition called sarcopenia, your skin loses elasticity, causing sagging and wrinkles, your hair may turn gray, and you may develop vision and hearing problems. And drops in testosterone and estrogen levels can cause erectile dysfunction and symptoms of menopause. Your body's NAD+ levels, which play a key role in mitochondria health, metabolism, and aging, start to decline significantly.
I should have eaten my vegetables.
I don't remember learning any of this in health class. This is also the time when you start gaining weight, which could increase your risk for diabetes or heart disease. In fact, the 40s are when men are most likely to develop heart disease. By the time you've reached middle age, your NAD+ levels are half that compared to when you were 20, and this can play a role in fat accumulation and worsening mitochondrial health. I'm saying this like I'm excited, but I'm really not. For women, the risk for heart disease is highest after reaching menopause, when your periods stop, which typically occurs between 45 and 55. Once you enter your 60s, your body is done making T cells, which are an important part of your immune system. This means you become more susceptible to disease. It takes longer to recover from them. You're also more likely to be diagnosed with cancer, as half of all cancer cases occur after 66. And this is about the time when the nerve impulses in your brain slow down, leading to a decline in some mental abilities like memory. But the real cognitive decline happens in your 70s and 80s. While symptoms of Alzheimer's disease can begin as early as your 60s, 33% of the population over 85 have Alzheimer's disease. The 70s and 80s is also when most people die from heart disease. By the time you're 80, you will have lost as much as 50% of your muscles compared to when you were 20. And speaking about death, the average life expectancy varies widely across the globe, depending on the health status of each country's population, with a low of 53 and a high of nearly 85. While aging is inevitable for you humans, even though we do have people working around the clock to help stop that, the best way to prepare for these changes and avoid major health issues is to eat well, get enough sleep, stay active and social, and see your doctor regularly. And no, searching your symptoms on Google doesn't count.
Heart is making weird noise. Oh God.
Other new Life Noggin videos discussed a brain-eating amoeba and breaking through from depression, and another answered some interesting questions from the internet. Find these on the Life Noggin YouTube channel. Also in March, Lifespan News released a number of videos diving into the latest in longevity science. Ryan and Emmett explored some surprising findings surrounding exercise, supplements, and protein, but also covered the potential brain-boosting effects of young blood. Here's more on that.
Heterochronic parabiosis is a well-known aging intervention in mouse studies, and it works. You stitch two mice together so they share blood, and like Highlander, the old mouse absorbs the life force of the younger mouse. It just happens to be one of the more, shall we say, unrealistic procedures for humans. I don't see a doctor prescribing you to stitch a toddler to your back for a few months to smooth out those wrinkles. Plus, that can't be good for your back. Welcome to Lifespan News. I'm Emmett Short. Today we're talking about the most problematic, yet perhaps one of the most promising cures for aging: young blood. New research published in Nature Aging has shown that factors in young blood can improve brain health this time. Studies also showed that factors in old blood, you guessed it, caused harm to young brains. That makes sense. I don't think anyone's surprised that old blood is not the cure for aging. In this study, they joined some 3 to 4 month old mice with some 20 to 22 month old mice. Mice of the same age were joined as well for a control group. The goal of this research was to analyze what effects heterochronic parabiosis has on multiple types of brain cells. See, other studies focus mainly on proteins or the proteome, but these researchers were looking at exosomes, lipids, and a bunch of other non-protein factors. Over 100,000 cells that consisted of 75 different types were analyzed. Similar to previous papers, they found multiple beneficial and rejuvenative effects on gene expression. The researchers found that 700 of the over 20,000 genes they analyzed had their expression changed. 442 gene expressions were changed in old animals by youthful blood, 155 gene expressions were changed in young animals by old blood. We don't know all the effects and impacts of these gene expressions and alterations, but based on what the study measured, the overall impact seems to be positive. So, in my view, that means young blood good, old blood bad. Some of the benefits were better gene expression, less senescence, improved intercellular communication that encouraged neurogenesis, the formation of new brain cells. One of the main things this paper highlights is the strong youth-promoting effects of parabiosis on the blood-brain barrier's endothelial cells. Endothelial cells have such direct and constant contact with blood, which may be why youthful blood has such a strong effect on those cells specifically. While not all the effects are known, including potential negative ones, here were 3 main takeaways from this paper. Youthful blood has a positive effect on blood vessels. Parabiosis could make the body process nutrients better, break down proteins more efficiently, and reduce age-related cell death. There may be new ways to slow brain aging and promote regeneration without targeting specific diseases or factors in the blood. I'd like to point out again, all these benefits are to the older mouse. I'm sure the younger mouse benefited from wisdom passed down from the older mouse during their time as conjoined twins, but medical benefits were not apparent. But what if it's possible to use a combination of youthful systemic factors along with blood scrubbing, known as plasmapheresis, to restore vasculature and function to the aging brain? These studies are helping us figure out some less ethically challenging and technically feasible ways to reverse aging. And more experimentation and eventual human trials will be needed to figure out if they work. And when those happen and when we have more to share, we'll have it for you right here. So make sure you subscribe to stay up to date. I'm Emmett Short, and we'll see you next time on Lifespan News.
Visit the Lifespan News YouTube channel to find all of their videos and more. That's it for this episode of the Rejuvenation Roundup podcast. Thank you very much for spending another month with us and for your help in the fight against age-related diseases. Whether you're donating, spreading the word, or simply listening to our content, we appreciate your help. Remember to subscribe, leave a review, and post about us on social media. This will increase our reach and introduce more people to the importance of life extension science. Don't forget, you can get additional deep dives into science, technology, and futurism on the Future Grind podcast. Find out more at futuregrind.org. Thank you for joining us, and we hope to see you next time on the Rejuvenation Roundup podcast.